Oh, hi there. Have you ever wondered how trillions of cells in our body have been functioning properly? Well, thanks to these four biological important molecules known as macromolecules, they have been keeping our cells in our body functioning properly. These four biological molecules are carbohydrates, proteins, lipids, and nucleic acids. First, we have carbohydrates. Carbohydrates are composed of three main elements: carbon, hydrogen, and oxygen. They store energy and provide the cell with instant energy. There are three types of carbohydrate: monosaccharide, disaccharide, and polysaccharides. Sugar and starch are some examples of carbohydrates. Next, we have proteins. Proteins are large chains of amino acids. There are 20 different types of amino acids used in our body. Our RNA carries the instruction to the ribosome on the order of the amino acid for each protein it makes. The structure of the protein depends on the sequence of the monomer, which is amino acid, and however it is constructed. Low energy hydrogen bonds between atoms in both polypeptide backbone and in amino acid side chains keep its unique shape. If a protein denatures, it loses its shape at any structural level. It may no longer be functional. In general, there are 20 different types of amino acid that are divided into two groups: non-essential amino acid and essential amino acids. Non-essential amino acid doesn't have to be part of our diet because our body can produce it. However, essential amino acid has to be part of our diet because our body does not produce it. There are four different types of protein structure: primary structure, secondary structure, tertiary structure, and quaternary structure. Next, we have lipids. Lipids are micromolecules composed of carbon, hydrogen, and oxygen, sometimes nitrogen and phosphorus, but fewer oxygen than other macromolecules. They have greater energy storage than carbohydrates, but their energy is not readily available, so they act as a long-term energy storage in the body because of their nonpolar and hydrophobic properties. Last but not least, we have nucleic acids. Nucleic acids are long chains of nucleotides. DNA is made up of adenine, cytosine, thymine, and guanine, whereas RNA is made up of uracil, cytosine, thymine, and guanine. Nucleic acids are important because they contain genetic information that has instruction for our cell growth, cell reproduction, and cell processes.
Hydrophobic and hydrophilic. These are two very unique chemical properties of molecules. These two dictate very much how well a molecule can handle and interact with water. So what I'm trying to say is, hydrophobic, as in the name is, does not like water and does not interact very much with water. It is normally non-polar. An example of hydrophobic molecules would be stuff like oil and wax that don't mix with water. It doesn't interact with water, it just separates itself completely from water. And hydrophilic, on the other hand, is the same thing but just the complete total opposite. Hydrophilic likes water and is normally polar. An example of hydrophilic molecules would be such as soap, because when you take a shower it mixes very well with water and it interacts, a lot of chemical reactions happen. And also sugar and salt, like when you mix it together with your tea. That is simply what hydrophilicity and hydrophobicity is.
Structure of phospholipid molecule. A phospholipid is an amphipathic molecule, which means it has both hydrophobic and a hydrophilic component. A single phospholipid molecule has a phosphate group on one end, called the head, and two side-by-side chains of fatty acid that make up the lipid tails. The phosphate group is negatively charged, making the head polar and hydrophilic, or, in other words, water-loving. The phosphate head are thus attractive to water molecules in their environment. The lipid tails, on the other hand, are uncharged, nonpolar, and hydrophobic, or in other words, water-fearing. A hydrophobic molecule repels and is repelled by water. Some lipid tails consist of saturated fatty acid and some contain unsaturated fatty acid. This combination adds to the fluidity of the tail that are constantly in motion. Phospholipids and biological membranes. The cell membrane consists of two adjacent layers of phospholipids which form a bilayer. The fatty acid tail of phospholipid face inside, away from water, whereas the phosphate head face the outward aqueous side. Since the head face outward, one layer is exposed to the interior of the cell and one layer is exposed to the exterior. As phosphate groups are polar and hydrophilic, they are attracted to water in the intercellular fluid. Because of phospholipids' chemical and physical characteristics, the lipid bilayer acts as a semi-permeable membrane. Only lipophilic solutions can easily pass the phospholipid bilayer. As a result, there are two distinct aqueous compartments on each side of the membrane. This separation is essential for many biological functions including cell communication and metabolism.
Have you ever wondered how water moves in and out of the cell? Well, Peter Azu just answered your question, as he was awarded as a Nobel Prize winner for discovering aquaporins. Aquaporins are basically water channels that are located in the cell membrane. They are responsible for the movement of waters in and out of the cell. They are located in the cell membrane that are made up of phospholipid bilayers.
Osmosis. Osmosis is the spontaneous net movement of water molecule from higher concentration to lower concentration down a concentration gradient. Here, the water molecule from outside is entering inside as well as water molecule from the inside is traveling outside. Here are the three tonicity of the cell: hypertonic, hypotonic, and isotonic.
In the first process, we'll be talking about hypertonicity. This is the process when the water potential of the outside the cell is lesser than that of the water potential inside the cell. This causes the water molecules to move out of the cell, leaving the cell flaccid. Yes.
In the next one, we'll be talking about hypotonic. This is when the water potential outside of the cell is higher than the water potential inside the cell. This causes the water to move inside the cell, causing the cell to be turgid.
In the third and the last process of tonicity, we have the isotonic process. This is the process when the water potential inside and out of the cell is the same, causing movement of H2O molecules in and out of the cell at the same rate. This creates no net movement.
Have you ever wondered when a dehydrated patient is admitted to the hospital, but somehow the nurses put them a drip that consists of salt and water? Well, why would they even need to put salt in when the aim is to just hydrate the patient with water? The answer is isotonic. Remember when we talked about hypertonic and hypotonic solutions that will cause the cell to burst or either go flaccid? Same thing applies to this situation. If the solution that goes into the patient's body contains purely water, it will cause the water to osmosis into the body cell, and this will cause the cells to burst. So in order for the patient to be hydrated properly, the solution that goes into the patient has to have the same concentration gradient of salt and water as the patient.